All right, guys. I'm gonna start the class today. My name is Alfredo Kirkwood, and I'm gonna tell you more or less everything I know about synaptic plasticity and how it's an important mechanism of memory. So it all starts. Excuse me. It's about how memories can be encoded in neural networks. Okay, there are at least three mechanisms that you can think about it. So you have a network of neurons, and if you change the electrical properties of the neurons, make them more excitable, for instance, eventually you're going to change the patterns of processing, and actually that could account for memory. The problem is that you have a limited capacity in that case, because a cell normally can fire between something like 0.1 hertz to 100 hertz, and basically you will have each cell probably will not be able to code to more than like a 10 or 20 different memories. The other possibility is morphology. So here we have a pyramidal cell in the hippocampus, and of course, if you prolong this dendrite or you shrink it, this dendrite where synapses are, of course you can change how the guy is connecting the inputs, and in principle you could you have no limit on how you can grow dendrites, and you may have a big capacity for storing memory. The problem is that they're too slow. Okay. So normally dendrites can grow at a rate of micros per minute at most, and memories can form in an instant. Okay, so you can have recollection of something that happened within a second. So the idea is a synaptic function, and that was something like actually Cajal, when he was first describing synapses, thought about that. So here you don't see that well, but you have all of these little, here one, here two, these little spines, to bulbophase, which are synapses, and a typical cell, a pyramidal cell in the hippocampus, which is very important for memory, but also the cortex, it will have like about 10,000 synapses. Okay. Now, if you think that there are about 10 to 11 cells, each one with 10,000 synapses, and then each synapse, you will see now, it may have like 100 different states. You have a large capacity, okay? And that is the idea, that memories are encoded as changes in the synaptic properties. Okay? And it's a relatively old idea. Most of what I'm going to talk today, it was envisioned basically by this very insightful guy, Donald Hebb, in the 50s, and he thought a simple way of encoding memory. Here you have an ensemble of neurons, all of them connected to each other, and then upon a presentation of a stimuli, this round thing, they get active. It doesn't show very well here the different color, but they get active, and the idea is that since they're interconnected, there's a reverberation in here, so they keep firing for a while. And as they are firing, the strength, the connection between them gets stronger. Now, eventually, you can present an incomplete form of the stimulus, and because all of the connections between them have been strengthened, now you can reenact basically the original pattern. And this is called pattern completion. And the only thing that you need to do that is that you have to have some mechanism that allow the connectivity between those neurons to get stronger. And he felt a very simple mechanism that actually is now true. People over the years have demonstrated that was true, and they're called helium synapses. And he thought, basically, if you have two axons, two cells, cell A and cell B, and cell A is connected to cell B, and make the cell B to fire action potentials consistently, okay, in an effective way, then that connection should get stronger. So now it's easy for cell A to make cell B fire. And that's called the helium postulate. And as I told you, you're going to see that this it holds many synapses. And So that was in 1950. It took almost like 20 years to somebody to think about the contrary. So if cell A is firing, but it doesn't make cell B to fire in a regular way, well, that synapse, that contact is ineffective and should be reduced, okay? And that was Gunther Stedt postulate. And basically, is the idea that positive correlation should be strengthened, negative or lack of correlation should be weakened, and that's the central tenet on current neuroscience, and then we will see how that happens. So I'm going to talk about these two mechanisms, long-term potentiation and long-term depression. And I'm going to talk about the induction, what are the signals? I mean, what is the type of activity that the axon A and axon B should have in order to get potentiation or depression? The expression means what is actually happening onto the synapse. Why the synapse gets stronger and why the connection gets weaker. And finally, the maintenance. And I don't think I'm going to talk that much about that, which is how you can make that synapse remain stronger for a long, long time. Okay? And here is a compilation of all of the research papers that have been produced over the years. I stopped about here. And this is the first long-term potentiation paper. That was in the 70s. That was actually 20 years after Hebb postulated the existence of this type of plasticity. It was demonstrated for the first time. And there was an exponential growth on that. And in all of the 80s, we learned about the induction, how the patterns of activity that can change the synapses. In the 90s and 2000, we learned about the expression, what happened to the synapse to get stronger or weaker. And now the big question is the maintenance. How you can, because what you really want to do is to have a change in the synapse that lasts a lifetime. Okay? And that's a big question because all of the elements on the synapses are exchanged basically in a week. All of those are proteins. Proteins are digested and content produced. So how you can maintain a memory in a single synapse. And that's a challenge that nobody has answered yet. Okay? So most what we learned about synaptic plasticity, it came from the hippocampus. Okay? So the hippocampus is part of the brain which is crucial for learning and memory. You may have heard about the HM, the poor guy that they removed the hippocampus and couldn't acquire new memories. And in the mouse or in the rat is like a little banana. It's a big banana, which is kind of like this. All right. And you have a good two properties. One, we know that it's involved in learning and memory. Without the hippocampus, you cannot acquire new memories. But it's also the gift of nature, or God, if you believe that, that given to neuroscientists. And the good thing is that you take this banana here and you chop it like a salami, and each of these slices conserve the basic circuitry of the hippocampus. So these are axons coming from the cortex, and they go to this thing called the dentate and these cells make contact with this other cells called CA3, and these cells make contact with all of this CA1, and this is the main at those synapses. Okay, and if you put here a big fat stimulating electrode, you know that the only thing that you're stimulating are those synapses. So that simplified the interpretation of the results. In cortex, you can also make slices of cortex, but you have a mesh of connections that you don't know what you are stimulating, you don't know what you're recording, and then you go exclusively to a particular cell. So it simplifies life. And here are experiments, okay? So in this experiment, what the people do is to put a stimulating electrode in here to give a little pulse, okay? And then record, in this case intracellularly, from a CA1 cell, and you get a response, okay? And here is time versus the magnitude of the response. They characterized by the slope, but you will ask, I will later. So those responses are very, stable. And then at this point, in these triangles, what they do is they give a tether. They stimulate, like 1 per minute or so, just to make sure that people are okay. And here, they give the tetanus, which is very high frequency stimulation, like 100 hertz. So they really shake those synapses, like, wow! And immediately, they get much bigger. Okay? So give from 100 to 300%, and it lasts. Okay? Now, if they repeat it, they give it more and more and more, and naturally it gets much bigger, and it lasts for 180 minutes. But that's more or less the most that you can usually record in one of these slice experiments. Okay? So that's long term. And that is the gist of memory, because you can have some memory that form very quickly. Normally, a traumatic memory. Something horrible happened, and I remember, yeah, I saw that, you know, like the typical joke I tell every year is that every animal in the forest know where they were when the Bambi's mother was shot. And it was so terrible that you know exactly what happened in there. And you remember that for the rest of your life. So here, these synapses, they knew that they were shot by this super tetanus, and they remain strong. Okay. Actually, the world record of long-term potentiation is held by this guy, Cliff Abraham in New Zealand. So he did the experiment not in a slice, but in a rat. He implanted the electrode, so he can record for many, many days. And actually, that LTP lasts for a year. Okay. So he gave it like a little buzz, okay, to those synapses, and those synapses remain stronger for a year. So that those synapses remember that they were buzzing. Okay. So this is kind of like the essence of memory. It's a good mechanism for memory because you can induce it very quickly, and it can last forever, basically. A year in the life of a rat is forever. Yeah, nobody in experimental rat. Okay, so I'm going to tell you now about a few properties that will make long-term potentiation so interesting. So first of all, as I told you, you want something with a big capacity. So you want all of these synapses to be controlled up or down independently. So if you do this LTP protocol, but all of the synapses in the cell go up, well, actually you have a capacity of 1, basically. So you want each one of these 10,000 to be independently, and they show it, okay? So what you're doing here, you stimulate 2 sets of axons, okay, into the same cell, go into the same cell, so pathway 1 and pathway 2, but to this one you give the famous 100 hertz tetanus, the super hertz tetanus, and that's the response before, and that's the response after. But the other pathway that was, non-touched, not overstimulated, it remained the same. So only synapses that are tetanized, they undergo this high frequency stimulation, can get potentiated. Okay? The next one is cooperativity. That's going to come interesting later. So people start to adjust the stimulation and say, okay, I'm going to stimulate 1 axon or 2 axons or 3 or 4 or 5 or whatever, okay, and see how many I need to produce long-term potentiation. And actually, you need 8. Okay? So in this particular case, okay, so you need a minimal amount. And that's something that's going to come in a few moments. That's going to be an important detail. So one single one, you stimulate, no matter how much you stimulate, you don't get potentiation. You have to be part of a pattern, okay? Part of a pattern of input activity on to convert it into the cell. A single one will not do it. And the third one, which is a combination of those, that's key, actually, that's beautiful, is associative, okay? So here we have one pathway, like say 8 or 10 axons, I just put one fatty one, okay? But that you're gonna stimulate with a high frequency stimulation. Well, that normally is gonna give you LTP. If this other pathway, you activate, but a low frequency, that normally doesn't give you LTP. But if you put now simultaneously this one with the high frequency stimulation, along with this one with the low frequency stimulation, this one can get LTP. So even if it was not stimulated on the high frequency, okay? So somehow this one can help this one to potentiate if they're coming together, all right? And that's so if you associate this one with this one, you can get potentiation here. And that's beautiful because this is more or less exactly what happened to the Pavlovian dogs. So you can replace this. This is the steak, okay, that's going to reduce potentiation no matter what. You're going to cause salivation. And this is the bell, the sound of the bell, that nobody doesn't do anything. But you put all of them together, now the bell can be potentiated and can code for something good. All right? So it tells you that this long-term potentiation can mimic or can reflect Pavlovian learning at a single cell level. Okay? So this is the minimal secret that it can produce Pavlovian learning is actually a single cell. So that's kind of cool. And because of those properties, everybody is very interested in it. So, and what is the mechanism? Basically, is this NMDA receptor. Are you familiar with the NMDA receptor? Everybody haven't heard about that? No? So, yes or no? No. You know? No. Okay. It's okay. So, this is a synapse. Okay? This is the presynaptic element. The action potential is coming from here and eventually reaching here and going to cause these vesicles to release glutamate. At least you know glutamate, okay? And this glutamate is going to act on the postsynaptic part, in the spine. Okay? And there are two types of receptors in here. In basically in every single brain of the mammal, etc., you have at least two. You have three, but actually two. The AMPO receptor, which is the classical one that allows you to pass sodium and potassium and the polarize the cell. Okay? And the NMDA receptor. Okay? And then NMDA receptor have a unique property, which is normally two properties actually. Normally it's blocked by magnesium. Okay, so it's a magnesium ion, very kind of like in the mouth of the channel, blocking it. Okay, so even if you bound the glutamate, this green thing here, doesn't happen because the magnesium is clogging the path of ions. But if these cells get depolarized, thanks to a lot of this AMPAs or maybe an action potential, then this part gets very positive, the magnesium gets out of there, and now ions can go through the NMDA receptor. Okay? So the NMDA receptor is going to fully function when two things happen. There is a presynaptic activity that for to release the glutamate to bind the NMDA, and there's a lot of postsynaptic activity to release the magnesium ion from the mouth of the channel. Okay, so those two things are important. So that is called coincidence detector, because basically the NMDA, it will be acting, it will be fully active when both the presynaptic element and the postsynaptic element are activated at the same time. And that's actually the postulate of Donald Heff in the 1950s, when axon A fires and makes axon B to be active in the same time, then that signal should be potentiated. Okay? So the NMDA receptor is a molecule that can fulfill that little computation, so to speak. Okay? Because it's a coincidence detector. It detects the coincidence of presynaptic activity and postsynaptic activity. All right? And of course, how people notice that is, well, if you block the NMDA receptor, okay, with an antagonist, with a drug, then you block the induction of long-term potentiation. Okay? And now, then you can realize now that the reason why you need the tetanus is actually the tetanus, the only thing that it does, the 100 hertz, is to depolarize the postsynaptic cell. So what happens when you give a single pulse, I'm gonna write it in here. Okay? Here is the response, anomaly decay. Now, if you give a second stimulus, but before the first response came, you add on top. Okay? And then a third one, so when you 100 hertz with this so frequently, you start to get bigger and bigger and bigger. And at some point, the depolarization is going to be so big that the famous magnesium block is going to go away. Okay? So that's the only reason why you need the high frequency stimulation. There's nothing about the high frequency per se, but it is as a way to depolarize the postsynaptic cell in an easy way. So if that is true, then, for no reason, then you don't need the high frequency stimulation. What you can do, if you can stimulate a low frequency, but then at this moment, you depolarize the cell. Okay? And you can depolarize the cell just simply by passing a positive current. Okay? So at this point, they depolarize the cell, they just make the cell very positive, okay? And in that case, the magnesium went out of the of NMDA. And when they stop the depolarization, the responses get bigger. Okay? So you don't actually you just need the depolarization, basically. Okay? And it turned out that LTP can induce by tetany and pairing low frequency, but with a positive depolarization. And of course, it's the same thing. If you give the tetanus now, which I tell you, the only reason the tetanus is to depolarize, and then you inject negative current during the tetanus, well, in that case, you block LTP. Okay? So you can block it by hyperpolarizing the cell during the tetanus. Okay? So that's it. And you can block it by blocking the famous NMDA receptor. An important thing is the only thing that the NMDA receptor does, and why it's so crucial, is allow calcium to go inside the cell. Yeah, here it is. You see? Calcium goes in. And that's important because it's calcium which eventually is going to produce LTP. All right? So the whole thing that you're worried now is to produce enough calcium in the postsynaptic cell. Okay? And so that I have now 20 minutes. So you can block it. I can tell you, if you put inside the cell a substance that will trap the calcium, these are called calcium chelator, well, you're not going to get LTP. And if you can deliver calcium by itself without activating NMDA receptor, without anything, you can also get LTP, and that's called by cage calcium. So what it is, it's a molecule, it's a big molecule, you know, that is trapping calcium inside, okay, and you have and then that molecule can be destroyed by UV light. So you flash you put that molecule with the calcium trapped inside the cell, through the electrode you're recording, and it's there, nothing happened, and then you flash UV light. The molecule is destroyed, and a lot of calcium is released. Okay, and in that case, you get also a potentiation of the response, just as you were activating an NMDA receptor, because you're bypassing all of those things. So the ultimate thing that you need is calcium inside the cell, and you can bypass that by putting those calcium. And what the calcium does, and I'm going to start here, what the calcium does is actually activating this protein kinase 2. Okay? And this is just the last part of this. Let me see. Yeah. So what the people do is they take this chem kinase 2, this is the enzyme that for it things, and they activate it. If you activate it, it's normally activated by calcium, but you can activate it in a dish. Okay? You have the activated one. You put it in the pipette that you're going to record, and then you record the cells with this active chem kinase 2. So what happened now, after chem kinase start to leak from the pipette, the recording pipette, into the cell, is they start to potentiate the synapses. Okay? So these are the responses. You get bigger. If you put the same kinase, but you boil it to kill it, nothing happens. All right? So. Okay. So the next question is how you can make the synapse stronger. Okay. So actually there are two ways. So in a synapse, basically you have the presynaptic component which is releasing vesicles. Okay. So one thing that you can do is to release more vesicles, for instance, and therefore you're going to have a bigger response. That's kind of obvious. And the other possibility is postsynaptic. You can put more receptors. Okay. Or you can change the properties of the receptors so they open more time. Okay, all kind of things. So actually during the 90s there was a big debate whether it was presynaptic or postsynaptic. Okay, now we know that it's postsynaptic. Okay, but during these heated days, people pre and post were arguing, and there was a very beautiful twist on the story that's kind of illustrative of how neuroscience progress. So I'm gonna dwell on that a little bit, which is the following. So, so people were doing all kind of presynaptic experiment and postsynaptic experiment. I'm not going to tell you, but there was one piece of evidence that for a while tilted the whole field to the presynaptic hypothesis. And it is the following. So are you familiar with the quantum release, no? No? No, okay. So the synapses are stochastics. So they can fail, basically. So if you're recording a postsynaptic cell and you're stimulating, sometimes you get responses, sometimes you don't get a response. And sometimes you get a response which is like this size, and sometimes twice, and sometimes three. So it's basically, the idea is that the release of vesicles from the presynaptic terminal is a stochastic thing, it's random. You have a probability of release. And sometimes you release nothing, sometimes you release 2 vesicles, sometimes 3, and sometimes 4. Okay? And that's complete, and you can describe that with a binomial distribution. Okay? So what happened now is that when you do that, you have to have only one axon and one cell. That would be a kind of very difficult experiment to do it. When you do that, and you measure before and after potentiated, when it was potentiated, this is what happened. Now you have bigger responses, of course, but the size of the individual things is the same. Okay. So, but now, before it was like you were releasing 1, 2, or 3, or 4 with a certain probability, and now it looks like you are releasing more, up to 6, for instance. And not only that, that the probability of failure is much reduced. Here, substantial time of the time, you fail to release, and now no. So it looked like it was presynaptic. So it basically, LTP is basically you're increasing the probability that you release vesicles when an action potential came to the presynaptic terminal. That was there. You follow that reasoning? Okay? So that was the end of the story, basically. It was cool, and everybody who was in the postsynaptic side, like my boss at the time, was very disappointed and kind of like, oh, our careers and stuff like that. Okay? Because the presynaptic guys win. But there was a very smart woman, Frances Edwards. I thought it was a guy. This is how they would do it. Okay, with that name. But it was a smart woman, and it just like Donald Hebb anticipates something, out of pure creativity. She thought, well, actually, it may look like it's a presynaptic failure. This one, but it may be postsynaptic failure. And she said, and here is a scheme. And she coined the notion of the silent synapse. Okay? And first of all, she noticed that all of these experiments for methodological reasons were done in very young tissues. So it's in slices from animals that are like 1 week old or something like that. And she thought, well, what if when a synapse is born, and that's the time when the synapse is being formed, at the beginning, they only contain NMDA receptors. Suppose that. Okay? So in that case, interestingly, if you release glutamate, well, these NMDA receptors can be blocked by the magnesium block that I told you. So unless you take a precaution that to have the postsynaptic cells polarized, you're not going to see it. Okay? Because normally, those experiments, for other reasons, they make it at a very hyperpolarized level. Okay? So if you're releasing, and that synapse only have NMDA receptor, you're not going to see anything. But now, if after LTP, now you incorporate AMPA receptors, now you're going to see a decrease of failure because now you're going to see the response. Okay? So this was a completely postsynaptic situation that it looked like presynaptic. Okay? So that is those failures are not presynaptic, they're postsynaptic. Okay? Think about that. Okay? And people think, and actually she was right. And people, first of all, they show when they start to stain those synapses at that point that it doesn't show very well in here, but you have to believe there is something in here. Okay, so there are, and there is something in here. There are basically synapses that only stain for NMDA receptors, and this is but not for AMPA receptors. Okay, so when you're very young, many of your synapses only stain for NMDA receptors. So that's the kind of like that type of evidence. And also, and this was a kind of like a more sophisticated experiment, and what they did in this, there might be a way to turn off the light on the front, I guess. But anyway, so this, it was the state of the art of imaging at that point. So they were using the two-photon microscope. You probably are familiar with the two-photon microscope. And this was one of the first applications of the two-photon. So what they're doing here is they have culture cells, pretty much better, now, in which they have transfected the cells with AMPA receptors that contain GFP, a fluorescent probe, in one of the things, so they can see it, okay? And here is the image. And now they give the tetanus, and 30 minutes after, what they start to see is that the manifestation, basically, of these AMPA receptors in the spine. These were not before, and now they're showing it there, okay? They have other things to show that those synapses were there before. So you have a synapse in here that it was before, it was devoid of AMPA receptors, and now you can see that fluorescent goes in there. So the fluorescent was kind of trapped in here, and then it goes in there. So basically, there's a mobilization of AMPA receptors from the dendrite into the synapse, okay? And that was LTP. Okay. Now, those experiments are not appropriate now because basically people say, well, look at this poor cell, you know, it's glutted with AMPA receptors. Cannot get any more AMPA receptors, kind of like choking on AMPA receptors. So, and here's a more sophisticated experiment, it's electrophysiology. So what they're doing here is they are recording at a very negative potential, minus 60, okay? So the NMDA receptors are not activated by then, so it's flat. And then they move the potential of the cell to plus 30. So it's very depolarized now. So now the NMDA receptors can conduct, and they conduct, okay? So before it's here, and now it's like that. So there are silent synapses, okay, that contain only NMDA receptors. And what they do is take one of these now, and then record at minus 60, like in here again, nothing happened. Then do the pairing at 30, so you activate the NMDA receptor for a while, very strongly, and then you come back to the minus 60, and now you get the responses. And those are AMPA responses. Okay? So the idea is that before in here, you have only NMDA receptors. During this pairing, you activate those NMDA receptors very well, and now you incorporate AMPA receptors, and now you can see it. Okay? So this is the idea now. The summary of LTP is basically that you have the NMDA receptors that are going to detect basically the pre and post synaptic coincidence. They allow to increase calcium, and that calcium will activate into this CAM kinase, and some stuff is going to happen. People don't know, or actually everybody knows, but everybody knows in a different way than the other guy. So it's a lot of fighting here. So this is the minimal model that everybody will agree. And as a result of that, AMPA receptors that were kind of sitting on the outside of the synapse, okay, that were not able to see the glutamine in here, now they move into there. Okay? So it's the lateral diffusion of the perisynaptic receptors that they go now into here, and that is LTP. Okay. Okay, so I have like 10 minutes to talk about LTD. All right. So it's depression. For a long time, LTP was the only game on town. Everybody knew that synapse can get stronger, but nobody paid attention that synapse also can get weaker, and also they need to be weaker for two reasons. One, if synapses can only get stronger, and anytime you learn something, you get stronger synapse, well, eventually, by the age 50, you know, your synapses are going to be like this big, and then you cannot learn anymore, you can explode. So you need also to reduce them now and then. But not only that, but there are many behavioral paradigms in which you need to decrease the gain of the synapses. For instance, and it's very simple. You can teach the rat, you know, food is going to be on the right. Right, this is my left. Okay. It's going to be on the left. And the right, after a while, then have to go to the left. But you can change the paradigm, and you can tell them now the food is, and the way to do it is probably you make LTP potentiate the synapses that are being conducive there. But then you change the paradigm and tell the rat, now the food is going to be on the other side. And the rat learn. So basically that's two things. Do LTP for this other direction, and it has to be depressed, the impulse to go to that direction. Okay? So you very often learning, it means shifting of preference, to potentiate a new preference and depress a previous preference. So in those cases, LTP, depression, and potentiation are equally important. It just happens to have in different synapses, but it's an equally important. So people were looking for a mechanism for depression, and actually they found it in the same synapses from CA3 to CA1. But in this case, the trick to depress them is to use prolonged low-frequency stimulation. So remember that for LTP, you have to do it like a high-frequency stimulation, or here like a lot of, and here kind of like a more long and actually, I was a postdoc in the lab when they discovered that. It was a student, and she was trying to get this LTD for almost like a year and couldn't get it. And one day she set it up to stimulate for 1 hertz for a short time, because we were thinking about, everybody was thinking about a short time. And then there was a seminar, and they have a pizza lunch. So she decided, okay, I'm gonna set this, I'm gonna, for 2 minutes, and I'm gonna grab my pizza and come back. And then she gets stuck and talking to somebody in the hallway. And when she came back 15 minutes later, oh crap, I give it 1 hertz for 15 minutes. And then she go back to basic stimulation, and she discovered that it was LTD. So it was completely serendipity, although she catch the thing right away in that moment. Okay, and now this is a standard protocol for inducing LTD. Okay. One of the surprise, it was like, okay, so if NMDA receptors are important for inducing LTP, they thought, maybe if I block the NMDA receptors, okay, and block LTP, I should get more LTD, okay? And they did the experiment, and actually, LTD was blocked too. So that was very amazing. So LTP and LTD both need the same NMDA receptor, okay? And that's a little bit counterintuitive. Why to going up I need to activate the NMDA receptor? Why to going down I need to activate the NMDA receptor? Okay? And actually, you also need calcium for the LTD. So to going up I need calcium. For going down I also need calcium. So the only difference with LTP, it was that you need phosphatases. So for LTP you need kinases. Somebody was going to put a phosphatase group with somebody else. And for LTD, you need a phosphatase, somebody who's going to take those phosphate away. Okay? So that was the only thing that, okay, something makes sense in here. Okay? So, so this is the minimal model of actually how bidirectional plasticity is coded in the synapses. And the idea is that the NMDA receptor is necessary for both, for potentiation and for depression. And calcium is necessary for both. What makes the difference though is the shape, is the dynamic, the kinetics of the calcium change. Okay? If you have a very brief and large change, like during this high frequency stimulation, okay, 100 hertz, you get like a big spike of calcium. That it will activate the kinases, particularly this one. Okay? Now, if you have a small and sustained calcium signal, you will activate phosphatase. And the important thing in here is that these phosphatase, they have a very high affinity for calcium. So just a little bit of calcium, you will activate the phosphatase. Okay? But more calcium will also activate the kinases, and the kinases are more stronger than the phosphatases. So a little bit of calcium, you only activate the phosphatase. More calcium, you will activate the kinases, and the phosphatase and the kinases will beat the crap out of the phosphatase. Okay, so that's more or less it. So, and that's how the same molecule and the same process, calcium inside the cell, can code for those. Oh, I finished. Oh no. All right. So I have 5 minutes to tell you about the $1 million question. That's an old question, now it's $1 billion, okay? Because $1 million, maybe not everybody, but it's not that difficult to get $1 billion. All right? So are these LTP and LTD involved in memory? Okay, so it works very nice in the slides, but do animals really use those? Okay, and there have been basically 3 approaches to check, whether LTP and LTD are actually engaged during learning. So one is obvious, you block LTP and you block memory acquisition. Are you familiar with the Morris maze? Yes, sir. Does anybody doesn't know about the Morris maze? The water maze? No? Oh, okay. So it was it's simple. So what you have here is a circular tank, and you put water in there, and then you drop a rat in there, okay? And you put what you do is you put a little platform submerged. So the rabbits have a lot of motivation to find the platform, okay? Otherwise it's sunk, all right? Okay, so it's very brutal actually, okay? But you don't have to do anything, you just drop it in there and then they will find it, okay? And they do. And so and it's great for quantifying memory because how you can check. So here is basically the escape, how long it takes to find the thing. And they get better, eventually reach asymptote. And the question is, how do you know that the guy really knows that this is there? Well, you nastily and secretly remove the platform and throw it in there, okay? And then the guy keeps looking in there, where is the damn platform, okay? So you can quantify, percentage of time they're spending here and stuff like that, all right? And these are experiments done in a mutant, okay? In these mutant mice, they eliminate the NMDA receptors from CD1, okay? And actually, as a consequence of that, they cannot have any ATP. But the NMDA receptors are everywhere fine, and everything else is fine. But they cannot have any ATP, so here the wild type and here the mutant. And the guy actually doesn't remember, kind of learn, but kind of like slow, and he doesn't remember where the platform is. Actually, I have an alternative hypothesis that the mutant is actually smarter, okay? Because they say, oh, there's no platform in here, maybe I'm going to look in another place, maybe there. But they don't like, when I talk to those researchers, they don't like that interpretation. They prefer that one, okay? So that is but it's basically, if you interfere with induction of LTP and LTD, you cannot code for new memories, okay? And this is it. So that's good. The number 2, and actually, that was a beautiful idea, is that anytime you learn, actually, some of the synapses are getting stronger, okay? And some of the synapses are getting weaker, okay? It's just like anytime you put something new in the computer, you're going to put some zeros in one place and ones in another one. So what happens to the computer if you move only once to the hard drive? You put only once, only zeros. Basically, you just reset the hard drive. You erase all of your memories. So if memories are coded on the differential potentiation and the pressure of particular synapses in different places, and you put all of them up, it's actually, you do massive LTP, basically what you're doing is erasing the memories, because all of the synapses are going to be at the top, okay? So actually, that's kind of like a cool and counterintuitive. They did it the first time that somebody said that they did it was actually kind of like a fishy, they didn't, okay? But the big problem in the hippocampus is that you have to induce LTP in all of the synapses of the hippocampus. And as I tell you, it's like a very long banana in the rat, and you have 2 of those. So Morris, the same guy who invented the water maze, he said, well, let's make things easier. So what they're going to do is they're going to remove one hippocampus, and the other hippocampus are going to reduce it like a third, just like a little tiny hippocampus, enough to put 2 electrodes. And some of those rats may learn in the water maze, and some of them learn. And what they learn in that puny little hippocampus, he did a massive RTP in that puny hippocampus, and then erased the memory. Okay, so they keep looking for the things. Okay, but the trick for that it was to reduce the size of the hippocampus. Okay, and the last one it would be that the formation of new memories actually. You can see as the animal is learning, some synapses are getting stronger, some other ones are getting weaker. Okay. Again, initially there was a false alarming here. People put the 2 electrodes, okay, implanted the electrode and put the animal in the water maze, to learn what the thing is. And they were saying, oh, cool, the responses are getting bigger. And actually the records of the responses in the CD1 were getting bigger. And it was also, yeah, learning a new place, the LTP, learning a memory, yeah, everything fit well. Except that Morris again say, hey, wait a minute, that's kind of fishy. So what he did, he decided to put a thermometer in the brain. And what happened, it was actually that as the poor rat was desperately trying to find the platform, it's swimming like crazy, and it's getting hot. And as they were getting hot, the responses were getting bigger because just a rain is basically just a big more temperature. Okay, so it wasn't that the guy was learning; it was just because they were getting hot. So they cooled down the water. Basically, they don't. Okay, there are ways now people can demonstrate that learning can increase the synaptic strength, but in more sophisticated arrays. And I think we're done. So basically, what it would take to show that, we need first of all very simple circuitry. So the hippocampus, even if it's cool for doing experiments and slices, it's still too complicated because you have many synapses. And you need to make very simple manipulations, okay? Killing receptors is not good because you produce all kind of compensatory things. So probably you will need to have a fast interpretation. But in general, people now will accept that long-term potentiation and long-term depression are the basis of memory. Okay? So 220, and we're done. Okay? Any questions? No? Okay.